Great. Thanks, Shoshana. Greetings from Philadelphia, where it is now 7 in the morning, and where we voted 80-20 to put President-elect Joseph Biden into office. So happy to join you all. This is a project that's been going on for more than 10 years in my lab to look at biological interfaces in crystals of protein structures and also assemblies. And we helped out with this, setting up this benchmark as Shoshana described. We can think of the biological assembly problem in crystals like this. When you download a structure from the PDB, it's the asymmetric unit. The asymmetric unit is the repeating unit that if you duplicated it, could make a model of the entire crystal, what crystallographers call an asymmetric unit. Symmetry operators will build up a unit cell like this, and then unit cells, when duplicated in 3 dimensions, in 3 directions, will build up a crystal. Now, somewhere in here is the biological assembly. It could be what's in the unit cell in this case, or it could be between unit cells if these were not symmetric objects. Some examples are down at the bottom. This is hemoglobin on the left-hand side. This is a A2B2 dimer of hemoglobin, and that's also the asymmetric unit of this crystal. But the one in the middle, the asymmetric unit, is actually just a heterodimer of the alpha and beta chains and is only half of the biological assembly. And the one on the right, the asymmetric unit, is actually 2 biological assemblies. Roughly half the time, the biological assembly, a bit more than half the time, biological assembly is the same as the asymmetric unit. About a quarter of the time, the biological assembly is larger than the asymmetric unit, like this one in the middle. And about a quarter of the time, the biological assembly is smaller than the asymmetric unit. So this is the problem. We need to look at a crystal and figure out, well, what's the correct biological assembly? And why would what were the purposes of doing that? One is to generate hypotheses of functional assemblies, if maybe a case where it's a weak dimer and we don't know what the correct assembly is. But also cleaning up annotations in the PDB. The PDB has annotations from authors and also from PISA. And the PDB-E actually marks a certain assembly as preferred with some data from QS Bio from Emmanuel's group. We think that these assemblies that are in the PDB are about 80 to 90% correct, depending on what subset you take. So we would like to be able to clean up some of those annotations. And then finally, evolutionary studies, looking at the same assembly in many different proteins within a protein family, and some proteins that have different assemblies depending on their functional states, modifications, et cetera. There are different approaches to this problem. In most cases, people assume that assemblies are symmetric and closed. That is, they're not sort of infinite chains. And of course, there are filamentous proteins like actin, but most protein structures in the PDB and crystals are closed assemblies, and usually symmetric if it's a homo-oligomer of some sort. Some of the approaches are to use biophysical features, like PISA uses surface areas and hydrophobicity and hydrogen bonds and things like that to assess what are good interfaces and what makes a good assembly. It's also possible to use sequence conservation. So EPIC from Jose Duarte uses sequence conservation within interfaces to build up assemblies to figure out what is the most likely biological assembly of a crystal. And then finally, a kind of bioinformatics approach, which is to use interfaces that are observed in multiple crystal forms of the same protein or of proteins in a single family. And also the same thing about assemblies. And this is the QS Bio approach and the approach that we've taken with ProtCID, which is the Protein Common Interface Database, which I'm going to talk about assemblies. So we started this by looking at a protein more than 15 years ago that was a sulfotransferase. And there was a paper in almost 20 years ago that this very small interface, if they did cross-linking and protease suggestion in mass spec, this small interface that was observed in one crystal at that time of this protein family was the biological dimer or the dimer in solution. At the time, got a colleague that was interested, we were looking at these, and I looked at all of the crystal forms that were available at that time in this protein family. So this is maybe 12, 14 years ago. And every single one of them had this dimer. Now, most crystallographers would think this dimer is too small. It's only 350 square angstroms. But it turns out, and subsequent experiments have shown, that it is the biological dimer. And so the idea of seeing the same interface in many different crystals of the same or family of proteins indicates may indicate that's the biologically relevant assembly or the biologically relevant interface. So we developed the ProtCID database to exploit this idea, and we kind of tested it statistically in 2008. But we developed this database to apply this principle to the entire PDB. And so the first step is to assign PFAMs. This is how we divide proteins into homologous groups. And so we do that with HH-certs, and we have a database called PDB-Fam. We build a 3x3x3 set of unit cells for each crystal so that we can get all of the interfaces of the asymmetric unit with other asymmetric units, both in the same unit cell and neighboring unit cells in each direction. We separate proteins in a particular family, in a particular PFAM architecture, into crystal forms. That is, if all of the interfaces in one crystal are the same as all the ones in another one, then it's the same crystal form. And so we count up the number of crystal forms. It's important to know how many independent experiments there are about interfaces. Sometimes people do 20 structures of the same protein in the same crystal form, and that's really only one experiment when it comes to interfaces, maybe different ligands, for instance. And then we cluster at both the chain level and the domain level. So clustering at the chain level is if we have a multi-domain architecture like this one, SH3, SH2, P kinase, we cluster all the interfaces of this protein with this protein. And then we also do it at the domain level where we break up every protein into domains and do all of the domains both within proteins and between proteins. So we would have SH3 homodimers, SH3, SH2, both within and between chains, and P kinase tyrosine with itself and with these other domains. So here's an example of the ACT domain, which is a regulatory domain in a lot of enzymes. And this is these are domain level interfaces. So on the upper left, we see ACT domain dimers. This is found in 32 different crystal forms and 37 different PDB entries. But it occurs, as we can see in all of these pictures, in many different contexts in multi-domain proteins, a bunch of different enzymes. So, but the blue-green dimer is basically the same in all of these structures, even it has all of these other domains attached. We use this with one of my colleagues, Eileen Jaffe, who was interested in phenylalanine hydroxylase, which the crystal structure that was available at that time had the ACT domains, these red domains that were quite separated from each other. But this was an inactive form of the enzyme. And so we hypothesized that this ACT domain interface occurs when phenylalanine binds as an allosteric regulator to this domain and forms a dimer. And later a structure of that dimer of the ACT domain has been determined. And so that helps us develop a hypothesis of how this particular enzyme family works. This is what the ACT domain webpage looks like for in ProtCID. And the page, basically, if you browse or you search by the PDB entry, you find a page like this that tells you there are 48 crystal forms that contain ACT domains. The first cluster has 35 crystal forms. That's 35 out of 48 is 73%, and 39 PDB entries. And then there's a list of all of the different PDB entries, their PFAM architectures, surface areas, what uniprotes they are, and pieces of information. If you click this number 1 here, you get a tar-g zip file with PDB files in it. And if you double click a PyMol script in there, you get an image like this that will give you all of the structures, and they're all listed by named by uniprot here. And so you can examine the interface. If you show the cartoon for the other domains is hidden right now, but those coordinates are present, so you could show all of the other domains as well. We use this to look at transient assemblies that maybe are not particularly clear experimentally. So an example of that is for RAS, for HRAS, KRAS, and NRAS. And there is a dimer that is present in 16 different crystal forms in about 100 and something PDB entries of KRAS, where it's the alpha 4 and alpha 5 helices that make a dimer interaction. KRAS, HRAS, and one structure of NRAS. The authors of this structure didn't note this dimer, and so this is not deposited in the PDB as an assembly. And many of these are also not deposited in the PDB as assemblies. There's now experimental data on all 3 systems that this is in fact a biologically relevant interaction of how RAS functions at the membrane. Also with RAS, here's RAS interacting with RA domain, RAS-associated domains. So a bunch of different proteins with different architectures, but this same interface is always the same between RAS and RA interacting domains. So this works for both heterodimers and for homodimers. In ProtCID, we also cluster not just protein-protein interactions, but protein-peptide interactions. So these are P kinase with, say, substrate proteins and other interacting peptides. And so you can look up any PFAM and look for different clusters of peptides bound to that PFAM. We also do Pfam-Ligand clusters. So this is pyruvate kinase. These are all the different ligands that bind, but we've also clustered those. And in the PyMol session that you download, you would see them. The substrate binding site, metal ions, ATP, ATP inhibitor binding site, activator binding site, bunch of different sites that exist in these proteins. And the same thing with nucleic acids. And so here's dsDNA with HLH domains, double-stranded RNA with DSRM domains, et cetera. And so you can look up a Pfam and look at how it interacts with nucleic acids. And in the last couple of years, we started to move on to thinking about assemblies. And so how do we do assemblies? Interfaces were easier, assemblies are harder. And the way we've done that is, again, we're looking at multiple crystal forms of homologous proteins. And we want to figure out what are all of the assemblies and how would we cluster them. So the first step is to generate all viable assemblies that cover a crystal. And we're using Jose Duarte's data from the EPIC website. And he derives assemblies that make up an entire crystal. They're all the symmetric assemblies. He doesn't do asymmetric ones. So that presents issues for certain kinds of proteins like filamentous proteins. But for most proteins, this works fine. So it generates all the homodimers, tetramers, any kind of symmetric oligomer or heterooligomers also. We sort the crystals by Pfam architecture. So we're always going to look at the same kind of length chain consisting of the same domains and assemblies made up of one or more different architectures. And we group them into crystal forms as we did with the interfaces. And then we're going to cluster the assemblies, all the ones that have the same Pfam architecture. So let's say P kinase plus cyclin, all those heterodimers. The same stoichiometry. So we're going to compare homodimers to homodimers, heterodimers to heterodimers, A2B2 tetramers to A2B2 tetramers, and having the same symmetry. So we're going to do, it's a C3 trimer, cyclic trimer. We're going to compare those to other C3 cyclic trimers across a bunch of entries or across a bunch of crystal forms. Same thing for D2 dihedral symmetry. And then we're going to assess these clusters, the number of crystal forms involved, the number of different uniprotes and their sequence identities. And then finally, also the number of crystal forms in the cluster compared to the number of crystal forms of the same uniprotes, of the same proteins, and their architectures across the whole PDB. The idea is if all of the structures of some particular architecture, all of the structures of a particular set of protein, a particular set of uniprotes, if all of those are in the cluster, that means that that protein or proteins always make the same assembly in every structure that's available. Now, that often won't be true because people make mutations or there are different ligands or different states of a protein where sometimes it makes an assembly and then sometimes it doesn't. And those, of course, could be interesting different functional states of the same protein. We need to figure out, well, how are we going to compare one assembly to another? Well, I've said we're going to do it by stoichiometry and by symmetry, but then how do we compare them? And what we're going to do is look at the interfaces within each assembly to compare one assembly to another. One way to classify interfaces for homodimers, for instance, is whether they're symmetric, isologous. So here's an isolate interface of BRAF kinase. Here's another one. So this crystal has this isologous interface and this one, or they're heterologous, they're asymmetric, like this interface down there. And the reason for doing that is in different symmetry assemblies, these are relevant whether the interfaces are isologous or heterologous. So to give you examples of that, if we're going to compare, a lot of structures are C2 homodimers, symmetric homodimers. And so comparing one homodimer to another one is just looking at the interface. Most of them are symmetric. And so we can look at this C2 homodimer of CSK and BRAF kinases, and they're the same interface. They're more likely to be the same biological assembly. In fact, this is the active dimer of BRAF. But here's another dimer of BRAF from a crystal, and it is also a C2 homodimer, but it is different. So we would cluster these as the same assembly, and this one as a different assembly. If we look at a C3 trimer at the bottom, this is in the OTCase family. These two assemblies are the same because there's a heterologous interface that makes up a C3 trimer. You've got monomer, monomer, and then monomer. And the interface that makes up this assembly that connects these three monomers is the same as the one in this one of these two different sequences. This third assembly, which is also in the same family, is also a C3 trimer, but it's a different interface. And so we would cluster these two as the same assembly and this one as a different assembly. If we look across the entire PDB and we do that for all of the proteins in this family, we get a cluster that looks like this. This is 55 different crystal forms and 38 different sequences, 38 different proteins. And this is the assembly that's observed, in all of the crystal forms that are available in the PDB of these proteins. This is for chloride dismutase. These are C5 assemblies, pentamers, cyclic. And if we compare the interfaces around here, we get this set of this cluster. This is 11 out of the 17 crystal forms that are available in the PDB. It's 11 out of 13 proteins in the PDB that have this assembly. However, all of the crystal forms of those 11 are contained in this cluster. The other two other sequences don't have this assembly. We can do this for larger, this is a cyclic trimer of trimers. So it's an A3B3C3 stoichiometry. And here's one C3 cluster. Here's the other two proteins, the gamma and beta subunits of urease. And this is 4 crystal forms and an A3B3C3 cyclic hetero nonomer, I guess. Dihedral symmetry is a little bit more complicated. This is a octamer, a D4 octamer of an enzyme called PBGS. Each cycle is, this is made up of 2 cycles, a C4 cycle, again, heterologous interfaces around the circle. Put 2 of those together kind of face to face, and you get a D4 octamer. The interfaces between the 2 cycles are all isomologous. They have to be symmetric. So this blue protein has a symmetric interface with the proteins in the other cycle. So in order to compare 2 different DN assemblies, we have to find one heterologous interface within each cycle and one isomologous interface. It really only takes one between one cycle and the other cycle, and then we have the same D4 octamer. Here are examples on the left of the PBGS octamers. This is 15 crystal forms of this same enzyme. These are the octamers. In the last remaining crystal form, there's actually a hexamer. I have a colleague that works on this. And this hexamer is an inactive form of this enzyme. So it's interesting to find a structure of one of these proteins that is not in the cluster because it's biologically interesting. This is IMPDH. It forms an octamer, and there are 17 different crystal forms of this octamer. But there's another octamer, and we were sort of wondering, well, what's this other octamer in these crystals that's in 5 crystal forms? Turns out this is a filamentous protein that actually makes an octamer like this orange one that binds to another octamer, this magenta one, and actually makes a filament. And you can see these filaments in cells. This is Ras binding to RBD. And remember, I showed you that Ras dimer. Here's a Ras tetramer where it's binding, this is RAF1 bound to HRAS. There are some caveats to this. There are filamentous assemblies. There are asymmetric assemblies. So this is an assembly that is an A2B. It's an uneven stoichiometry. It's a homodimer bound to a 3rd protein. And EPIC doesn't typically generate these correctly. And there are assemblies that are asymmetric, like EGFR. This is an asymmetric dimer, homodimer, and but it's not a filamentous structure, and also EPIC doesn't generate these. And finally, some crystals can be mixes of assemblies. This is P53 bound to DNA, but only one of these is bound correctly to DNA, and the other ones are not. And it's sort of an assembly of maybe 1 or 2 proteins bound to DNA, and then a 3rd one that's kind of extra. Given that, I'm almost done. We can think about, well, could we fix up the assemblies in the PDB? And if we look at all the clusters of multimeric assemblies that we found with at least 3 different crystal forms, so we're not looking at monomeric proteins. And if we look at the ones that EPIC thinks are that 50% of the assemblies in the cluster are biological, that is, these are likely to be correct assemblies, that's about 47,000 entries. The PDB only contains 38,000 or so of those. So that means only about 81% of the entries in the PDB have what we think may be the correct assembly. That means 19% are missing. So there's a lot, this is a way to identify a lot of extra assemblies that may be correct and that we could add to PDB annotations. And I just want to acknowledge this is all really work by Qifeng Xu, who's a senior programmer in my lab and has been in my lab for about 14 years. And these are the 2 websites. And I want to thank you for listening.